I hope you all had a nice break. And we have more fantastic speakers lined up for the afternoon session. I'm really looking forward to that. It's my pleasure to introduce the first speaker, Robert Noble, who actually just left the lab of one of our contra PI, Nico's lab. Dr. Noble obtained a DPhil in zoology from the University of Oxford in 2014 with a thesis on mathematical modeling of infectious disease. He then held postdoc research position in mathematical oncology in France, moved on to ETH Zurich, and then also the University of Zurich. He then joined City University of London as a lecturer in July 2020. So welcome, Robert, coming to join us in this on this little island. And the topic of his talk today is on how tumor ecology shapes evolutionary dynamics. Thank you, Robert. Over to you.
Thanks very much, Yin Yin. I will now try to share my screen. Okay, it seems to be working now. Okay, I'm sorry about that. It's a brand new laptop, and so there are one or two things that... There we go. Are you able to see the presentation now?
Yes.
Excellent. Okay, so yeah, thank you very much for inviting me, and thank you, Yin Yin, for the kind introduction. What I'm going to talk to you about today is a project on spatial models for forecasting tumor progression and survival. And this is work that I did. I started this, in fact, when I was in Montpellier, and I continued it in Switzerland, and I finished it just recently. So as Yin Yin said, I'm now based in London, but my background prior to that, I started off, in fact, in pure mathematics at the University of York, where I did an M-Math degree, largely in algebra and number theory and not very applied mathematics. I then spent a few years doing other things, in particular working in communications, and I got interested in science communication. And that really led me back to reading a lot of literature, and particularly mathematical modeling literature in infectious disease. And I started realizing, well, maybe I could be writing these papers rather than just reading them and interpreting them for the public. So I made some applications for a PhD, and then I ended up in the zoology department in Oxford, where I studied infectious disease modeling, and in particular, the immune evasion dynamics of the Plasmodium falciparum malaria parasite, which are very complicated and required me to learn what I later discovered are really more computational biology techniques, as well as mathematical biology. After that, I decided to switch to cancer evolution research, and that moved me to Montpellier, where I got involved in modeling cancer at every different biological level, going from cells right up to organisms. And following that, I decided that I wanted to make more use of the molecular data that's so abundant in cancer. And so I took a second postdoc at ETH Zurich with Nico Bierenwinkel in a computational biology group, where I got more into modeling somatic evolution and genetic evolution. And simultaneously, I took on a 20% of my contract at the University of Zurich with Hannah Coco, where I pursued more evolutionary and ecological theory, looking more at organismal evolution. So all of these different parts, how do they add together? Well, they add together in what we call mathematical oncology, which incorporates all of these methods in one framework to understand what's going on in tumors. And my particular interest really is in evolutionary processes and how they're shaped by ecology. So in the talk, in the questions afterwards, I'd be happy to take questions about this career path and whether you think it was a bit meandering or whether it makes sense in retrospect. So as inspiration for this project, here is a paper from a few years ago. The first author is Carlo Meli, but there are many familiar names on this author list. And the purpose of this paper was to propose that tumors can be classified by quantifying their evolutionary and ecological features. So what you see, for example, here is what they call an evolutionary index, which is composed of two factors corresponding to the amount of heterogeneity at one point in time, the spatial heterogeneity, and the change in that heterogeneity, so the turnover in heterogeneity. And these two factors lead to four possible classifications, four different types of tumors. And similarly, this paper proposes an ecological index, and then we end up with very many possible types. Now, what the authors propose is that this way of classifying tumors can help us to understand how better to treat and forecast the development of tumors. But within this paper, this is really a conceptual framework. There's no actual modeling within this paper. So one of the things I wanted to do was investigate how would this work out? How is this consistent with an actual mechanistic evolutionary model? Another inspiration for this study was the TRaining renal cohort study, which is a fantastic clinical trial. One of the main, in fact, the main primary outcome measure of this study, pre-registered, was to validate an intratumor heterogeneity index, what they call an ITH index, and a measure of genome instability, what they call WGII, as prognostic markers of progression-free survival. Here it says these should be stage and grade independent. So in other words, these should be better than the status quo of evaluating tumor prognosis based on stage and grade and conventional systems. The idea is that intratumor heterogeneity and whole genome instability should be able to add more information than one gets from the conventional approaches. So the outcome of that study, or at least a preliminary outcome from the first 100 patients, which was published a couple of years ago, finds that, well, they're kind of mixed results. So if we concentrate on the top row here and the top left, these are results from the TRACERx renal cohort, the first 100 patients. They will recruit and report on more patients, so these results might change. But to begin with, what they find is that when they look at their measure of diversity, their ITH index, they find that indeed the progression-free survival is lower for patients with a high degree of intratumor heterogeneity overall. And here they've just split the patients into two groups, high and low, either side of the median. So the yellow line is high intratumor heterogeneity and the black line is low. And they find that, yes, this is a good predictor of progression-free survival. The second plot in the top row shows that the same thing happens for genome instability, that patients with high genome instability tend to do worse than those with low. And the final plot in the top row shows what happens if you can combine these to split the patients into four groups. And there you find that the best outcomes are for patients who have low intratumor heterogeneity and low genome instability. Among the other groups, it's a little hard to tell what's going on. I think that might become clear as they add more patients. And in the bottom row, you see what happens when they apply the same analysis to TCGA data. They found the same kind of pattern in an independent, bigger cohort. The advantage of the TRACERx renal cohort is that they had multi-region sequencing and they had some very sophisticated techniques for measuring intratumor heterogeneity. The Cancer Genome Atlas data set is larger, but is generally only single sample bulk sequencing. So there are pros and cons here. But what you might notice is that you might just be able to see the p-values here, and where it says p-value adjusted on this top set of plots, you see that the adjusted p-value is not significant. And this is an adjustment after adjusting for stage and grade. So in other words, these measures were useful, but they didn't outperform the status quo, at least within their cohort. When you look at the larger TCGA cohort, they find that it does outperform it. So there is hope that if you have a big enough cohort, maybe. So the question then is, why does this kind of pattern arise? Why is it that higher heterogeneity leads to worse outcomes? So in order to test this, I developed a stochastic agent-based model of solid tumor evolution. And this includes a form of spatial structure. We assume that, we model cells as being placed on a regular grid of sites, which we call demes. Each of these sites or demes can contain multiple cells. And we assume that the cells within the same deme are all interacting with each other and competing. And there's a carrying capacity, a fixed maximum population size for each of these sites or deans. And we assume that the tumor grows by invading healthy tissue. So we start with a grid that is filled with healthy cells, and we introduce one transformed cell to the middle of the grid. This transformed cell has a slightly higher proliferation rate than the normal cells, so it tends to proliferate faster and replace the healthy tissue, and then it can invade neighboring sites and so spread through the normal tissue. And we allow this tumor to accumulate driver mutations stochastically, and these mutations increase the cell division rate multiplicatively. So the kind of results we get, if you look at the top right of this figure, you see the spatial layout. This is just a two-dimensional tumor. We model two-dimensional cross-sections through larger tumors, essentially. And each pixel in that kind of pixelated plot in the top right represents one of these deans containing hundreds of individual tumor cells. And I'm not showing the normal cells, but this tumor is, in fact, within the model, this tumor is surrounded by normal tissue that it's having to invade. And the different colors there correspond to different combinations of driver mutations. So different clones defined in terms of driver mutations. And on the top left, we see the corresponding frequency dynamics over time. This is called a Muller plot. So the original tumor cell belonged to the grayish brown clone, which is shown from generation 0. And then within that original population, a light blue clone emerges, and that spreads within the tumor. And then that gives rise to further clones with different colors, as you see in that diagram. And generation here is the number of cell divisions relative to the initial tumor cell. In the middle here, we see the population, that the population is expanding. And at the bottom, we see the same information, but this time, instead of coloring by the genotype of the clone, we color by its division rate, so its fitness. And you see that the fitness increases over time, and that the fittest clones spread within the less fit clones, as one would expect. And in this example, we end up with a lot of heterogeneity building up over time. So in order to parameterize this model, well, some of these parameters aren't very well known, and therefore we considered a wide range of parameters. For the driver mutation rate, we considered values between 10 to the minus 6 and 10 to the minus 4 per cell division. Often modeling studies assume something around 10 to the minus 5 based on various forms of evidence. So what we're doing here is we're allowing an order of magnitude in either direction. The mean driver fitness effect, so each driver mutation, when it occurs, the effect of that driver mutation is chosen from an exponential distribution, so that not all the driver mutations have exactly the same effect. Some of them can be bigger than others. And for the mean of that distribution, we consider values between 5% and 20%. So these, when a driver mutation occurs, it will increase the proliferation rate of the cell it occurs in from anything between, on average, anything between 5% and 20%. And these values hover around values that have been inferred from solid tumor evolution. And for the number of cells per dean, this is essentially how many cells is the typical cell interacting with within a tumor. And we really don't know this parameter, unfortunately. It turns out to be a very interesting parameter, but it's a very difficult thing to measure. So again, we allow for 2 orders of magnitude, anything between 64 and 4,000 or so cells. And finally, we set the dispersal rate, the rate at which these cells disperse from one dean to another, depending on the other parameter values. So that's not an independent parameter, it's a dependent parameter. And we set that so that all of these tumors, regardless of their other parameter values, they all take a few hundred, something around a few hundred or 1,000 cell generations to reach 1 million cells, which will correspond to a few years of real time. And this is consistent with the best estimates we have for the number of cell generations it takes for a tumor to grow. So what we get then is, if we run this model, if we run, say, 100 instances of this model with exactly the same parameter values, then we end up with a lot of growth trajectories, as you can see here. And what I've done is I've just adjusted these growth trajectories so that they all intersect at a particular intermediate time. So I've just repositioned them along the x-axis so that they all intersect at this point, which I've labeled time 0. And so what we do is we observe these tumors at what I call the measurement time 0 here. And this is the time when the tumor reaches a fixed measurement size. So basically, we grow 100 simulated tumors, and then we record the time it took them to grow to the measurement size, and we define that as time 0. We observe the tumor at this time 0, and then we use those observations to try and predict what this tumor is going to do next. And we then measure how accurately we were able to predict. So in this example, I've colored these trajectories according to the mean cell division rate at the time of measurement. And one would expect that tumors that have higher mean cell division rate at this early time point would tend to grow faster in future. And indeed, that's what you see. The red ones, which have high cell division rate, tend to grow faster than the blue ones, which have low cell division rate. And therefore, if we look at a correlation between the cell division rate recorded at this time 0 and the time it took to reach an endpoint size, shown at the top here, we would expect a correlation between the cell division rate and this measurement, and this delta t, this time difference, the waiting time until the tumor reaches its final size. So we're getting from this a correlation coefficient, which in this case would clearly be close to 1 for a good correlation. And we want to know, well, how good are these correlations in different circumstances? So these correlation coefficients are what we really care about. And if we look at cohorts of tumors with identical parameter values, as I showed you in the previous slide there, what we find is indeed, if we look at mean cell division rate at a certain tumor size, and here I show for various different tumor sizes, and in the paper we look at all kinds of other parameter values and scenarios as well. But in general, we find that this mean cell division rate is a very good predictor of how quickly the tumor will continue to grow. And even if we measure very, very early on, and we measure how long it takes the tumor to reach a much larger size, still measurements you make very early on have a lot of prognostic value. But if we look at clonal diversity, a measure of intertumor heterogeneity similar to what TRACER-X did, we find that if you measure early on, yes, it's a pretty good predictor. But if you measure later on, it can in fact correlate in the opposite direction in some cases. So we find that tumors that are more diverse actually grow slower than ones that are less diverse, depending on when you measure and various parameter values. So overall, this is an unreliable measure. It depends on too many things. But just to remind you, all of these tumors within the cohort here from which we're calculating the correlation coefficients, they have identical underlying biological parameters. So all the differences between the development of these tumors are entirely by chance. So that makes it quite hard to forecast, in fact. And so why is it that higher diversity can correlate with slower tumor growth? Well, if we look at an example, if we look at a particular tumor that within a cohort here, so each dot here is a tumor within a cohort with identical biological parameters. If we look at a case which has low clonal diversity at the time of measurement and high cell division rate, then what we see is that the reason this tumor was able to achieve a high cell division rate and low intertumor heterogeneity is because it had many selective sweeps. And you can see that its division rate evolved quickly by these selective sweeps. If we look at a case here, this tumor has the opposite. It has high clonal diversity and low cell division rate. And we see that's because there's a lot of clonal interference. Just by chance, this one didn't acquire a strong driver early on. Instead, it acquired many weaker drivers later on, and all of these had similar fitness, so they compete with each other and interfere, and this slows down the growth of the tumor. So we see that, in fact, the rate of clonal turnover matters, as well as the amount of intertumor heterogeneity. And again, this is just for tumors which have identical biological parameters. We can get these different outcomes. And if we look across many different parameter values, so here we've varied the driver mutation rate, the driver fitness, mean driver fitness value, and the Deem size, which is the value of K here across the panels. What we see in general is, again, we can see these negative correlation coefficients between clonal diversity and future growth rate. In many cases, we find this negative correlation. And specifically, we find it in cases of tumors which have a high degree of clonal turnover. But in reality, of course, tumors vary in their biological parameters. And in particular, as we saw in the TRACERx study, they can vary in terms of the extent of genomic instability. So therefore, what we try doing is, instead of keeping the biological parameters fixed within each cohort, we simulate cohorts in which the driver mutation rate is a random biological parameter that can vary between tumors. And in this case, we find that the correlation between our predictor variables, such as clonal diversity, and the future growth rate is reliably positive. And this is because, in fact, the amount of diversity is acting largely as a proxy for the underlying amount of genomic instability, or the underlying driver mutation rate. And this is robust to multi-region sequencing. So here we simulated what happens if we take multi-region sequencing and we just calculate diversity based on those samples rather than across the entire tumor. And the result stands up. So finally, what we did is we added resistance mutations, because so far, all we've done is we've tried to predict how rapidly a tumor will grow after we observe it. But that's rarely what we want to do in the clinic. What we really care about is, how is this tumor going to respond to treatment? Usually, if a large tumor is detected, it is then treated, and we want to know, well, what's progression-free survival after treating the tumor? And in fact, that was the end point of the TRACERx study. So what we do then is we add to the model the occurrence of resistance mutations, and so that not all of these tumors acquire resistance mutations, so that, in other words, so that some of them are curable, we assume a low rate of mutation for resistance, and we assume it's 10% of the driver mutation rate. So it's still linked to the amount of genomic instability. And these resistance mutations are neutral prior to treatment. When we apply treatment to the tumor, it kills all of the cells that lack the resistance mutation, whereas those with the resistance mutation can grow back. And so in this case, we see results like shown in the top row here. So we allow the tumor to grow. In this case, it grows for about 1,200 generations or so, until it reaches an endpoint size. At that point, at the measurement time, we treat it, and all of the non-resistant cells are killed off. And then the tumor rapidly regrows. And the reason it rapidly regrows is because there is less normal tissue. So the tumor is debulked, and then the surviving cells grow essentially into more or less empty space where all the debulked tumor used to be. So this accounts for very rapid relapse, which is similar to what's seen in the clinic. The relapse is faster than the growth of the original tumor. And so what we do then is we measure the amount of intratumor heterogeneity just before we treat, and then we see how long it takes the tumor to regain its previous size. And this is a proxy for progression-free survival. It's not exactly progression-free survival, but we think it's a reasonable proxy that will correlate with actual progression-free survival. And what we see is that when we look at the correlation between the intratumor heterogeneity measured just before treatment versus how long it takes for the tumor to regrow to its pre-treatment size, we see consistent negative correlations, which is what we'd expect. And these are consistently negative across different parameter values. So in other words, the results we saw for tumor growth, they translate to forecasting progression-free survival quite straightforwardly, as we'd expect them to. And if we look at that in terms of Kaplan-Meier plots, in the top row here, you see the results from the simulation, where again, we've split the patients into two groups with either high or low clonal diversity and high or low driver mutation rates, just on either side of the median. That's in the first two panels, and in the final panel, we've split them into four groups for the 2x2 combinations of clonal diversity and driver mutation rate. And the coloring here is similar to what was used in the TRACERx study. So in the bottom row, you see the TRACERx results that I showed you previously, and in the top row are simulation results. And you can see that there is some consistency there, that we also find independent correlations with both clonal diversity or intratumor heterogeneity and driver mutation rate or genomic instability. And when we do the 2x2 comparison, the best results are in the patients that have low intratumor heterogeneity and low genome instability. And the reason they did this in the TRACERx study, they split into groups like this because their measures were not particularly precise. But of course, we know precisely what these values are. So we can also look at a continuous measure, and we can look at a Cox proportional hazards model in which we have diversity as a continuous predictor term. And in that case, again, by itself, we find that there's a very good low p-value. It's a very good predictor as a continuous predictor. But if we have a model in which we include diversity D and the driver mutation rate mu together, then we find actually the driver mutation rate is a better predictor. It's significant, and the diversity doesn't really add anything extra to it. And the third term here is just the fact that the predictive value of the mutation rate varies with time as well. So we did various versions of these models in the paper, and the main conclusion here is that if the mutation rate here is something that can be taken into account in conventional measures of stage and grade, then this would explain why intratumor heterogeneity by itself doesn't add a lot of value. If you have a lot of variation in the biological parameters, then diversity by itself across the entire tumor, across the entire cohort, doesn't add very much. You have to be a bit more subtle about how you analyze than that. And this seems consistent with what they found in the TRACERx study. So in conclusion, what we find is that this kind of eco-evolutionary modeling can explain clinical observations. It can shed light on possible explanations for those observations, as we've seen here in the case of TRACERx. And we've seen that appropriate modeling of spatial structure in particular is essential for characterizing and forecasting and indeed controlling tumor evolution. So we found that depending on the underlying biological parameters, we could get different predictions. And I've developed this more in a companion paper, which is currently a preprint. And with regard to this proposed method of categorizing tumors according to eco-evo biomarkers, well, this shows promise, but one has to be very careful about how one interprets these different factors and consider interactions such as the fact that that a high degree of intratumor heterogeneity has different interpretations depending on whether there's a high or low degree of clonal turnover, which, to be fair, is something that was discussed in that paper I showed you earlier. So to wrap up, I'd just like to thank the people who worked with me on this project and on related projects. So this work started with Mike Hochberg and John Burley in Montpellier. So Mike was my first postdoc supervisor, and John was a visiting me master's student. And so, in fact, the genesis of this project was his first year master's thesis. He's now a PhD student at Brown. And this work was particularly moved forward recently by Cécile Lesueur, who was visiting us as an intern at ETH and has now started a PhD at EMBL Heidelberg. And she ran a lot of these, the models that the results of which you have seen in this presentation. And I'd also like to thank everyone in the Bierenwinkel group, particularly Nico, Dominic, and Jeanne, who moved forward related ideas and related projects, and my collaborators Jakob and Yannick for a lot of useful advice and also contributions to related projects. So the paper, if you want to read it, is in Evolutionary Applications, published earlier this year. Thank you very much.
Thank you very much, Rob, for a fantastic talk. Questions, please.
Yeah, I have a question.
Yes, please.
Yeah, thanks for the wonderful talk. It's related with my project, my PhD project, estimating growth rate. I wanted to ask you something about you mentioned that the mutation rate is varying over time. So you have a base mutation rate, and then you have multiplicative variations of that mutation rate across the clones, right?
Not quite, no. So the proliferation rate, the proliferation rate increases every time a clone acquires a driver mutation, but that we don't look at evolution of the mutation rate. So the mutation rate is a fixed parameter. I think it will be interesting to look at evolution of the mutation rate, but I think that is a kind of a project in itself. So maybe I think I poorly explained the Cox proportional hazards model at the end. What happens there is that even though the mutation rate is fixed, the effect of the mutation rate on outcomes is time dependent. So this means that tumors that have high mutation rate, there's a different influence on the prognostic, on the progression-free survival, depending on whether that tumor has grown for a short time or a long time. So that really, this is just kind of a technical detail. We looked at models with and without that factor, but technically one should include it because it allows us to satisfy the statistical assumptions of the test, essentially. So I think there are some subtle, interesting reasons for why that arises, but I haven't looked at them in detail.
But sorry, related question about just a little quick. Related with that, if proliferation rate changes across over time, then if mutation rate is constant, is fixed, but is the mutation rate is per cell division. If you increase the cell, if you increase the cell proliferation rate, then you also increase the mutation rate.
That is true. Yeah.
So yeah. So I'm looking forward to see the details in your paper, but it's really interesting. It's really related with my project.
So that this is an important point, that yes, we assume that mutations occur during cell division, and therefore the clones with higher proliferation rate also evolve faster. And in fact, this can give rise to Darwinian demons, because one can end up in the situation where these cells are acquiring more and more drivers, and so they're going faster and faster, and they get more and more mutations. The way we got around that, because this is partly a technical problem, that the model will then explode, right? Because it becomes infinitely fast. But it's also not biologically realistic to assume that proliferation rates can increase forever. So what we do is we take inspiration from the work of Richard Lenski and colleagues, where they found that, at least in E. coli, there's diminishing returns epistasis. So although fitness increases over time, the returns from each additional beneficial mutation get smaller, progressively smaller. And so we used a function a bit like that, assuming that there is eventually some limit to how fast these cells can proliferate. And so the closer they get to that limit, the smaller the driver mutation effects tend to be. So that seemed like a reasonable approximation to what we assume is happening in tumors. They can't get faster forever.
Okay, thanks a lot.
That's great. Any more questions?
Hi, thanks for the really nice talk. I wanted to ask a question which is actually related to what you said as an answer to your last question, the last part. So maybe there's some number of mutations that doesn't really give additional evolutionary advantage, and that maybe even makes the genome completely unstable and maybe even kills cancer cells. And also something that it is also connected to something that I've noticed on your Kaplan-Meier curves, where you have been comparing your survival results to the TCGA results, and it looks like your patients die more than real patients. And I was wondering, why is that? Maybe there's some additional parameter you can add up to your model that will actually act not in favor of tumor, but against tumor.
Thank you. Yeah, these are good questions. So the first question about deleterious mutations, this is something we definitely considered. And in fact, when I started playing around with these models, one of my original intentions was to look at deleterious mutations and how they accumulate and so on. It turns out that this this project was complicated enough, and the patterns we found were complicated enough without also including deleterious mutations. But I think it would be an interesting continuation of this work to include both mildly deleterious mutations that can build up because they aren't efficiently eliminated, and also more seriously deleterious mutations such as you suggested. As I say, the reason we haven't done this so far is simply because we already had enough of a complicated story to tell just what happens when one uses the simplest possible imaginable model, which only has beneficial mutations. It's already fairly sophisticated, the kind of patterns that can emerge. And if one starts adding deleterious mutations onto that, I think that needs to be done as an extension. But I think it is interesting because what it would do is it would increase a cost of high genome instability, which currently we don't have. So currently high genome instability in our model just means that tumors more rapidly acquire driver mutations, and that's beneficial to their growth. But one could imagine having some kind of trade-off where deleterious mutations were also present. The problem then is, well, how does one parameterize that trade-off? And there are a lot of design decisions to be made there. And that's why I think it's a project in its own right, really.
Yes.
The second question about the Kaplan-Meier plots. Yeah, I didn't change the parameters or tune the parameters such that we would get exact quantitative agreement between our results and TracerX. What I was more interested in doing was just revealing a qualitative pattern that gives us some insight. I think it would be possible by adjusting things like the resistance mutation rate in particular, to make it so that more of these tumors are cured. Because if we make the resistance mutation rate lower, then more tumors will fail to acquire any resistance mutations, and more of them will be cured, and therefore outcomes will be better. So that would be quite an easy thing to do, but the way I decided to do it was not to kind of fiddle around too much and just go for a simple assumption. Well, let's make it just 10% of the driver mutation rate, and we get something that's already qualitatively consistent. Because I think aiming for something that's exactly quantitatively consistent is a bit artificial because there are so many unknown parameters here.
Yes, that's true. Okay, now I understand. Thank you.
Is there time for another question?
Yes, please.
Yeah, I think it's also, I wanted to follow up on Magda. I think we have very similar ideas. So I think another aspect that would, make it even more interesting and even more complicated, but also very, very exciting would be to investigate epistatic effects between mutations, right? So there's one thing of this deleterious. I think also Magda probably actually had in mind these potential interactions between mutations, and which can act in both ways, right? So they can be, let's say, worse for the tumor or better for the tumor. And following on this, actually also about this, like, let's say, how to explain different things in your model. I think it seems like you could eventually, like, pretty quickly go into problems of identifiability of, or problems of like actual causal factors, because you could say, I think you were saying, oh, I could play around with the resistant mutations and so on, and then the patients would die similarly, like in the TracerX. Probably Magda had an idea, if you add additional mutations that kill the tumor, the tumor slows down growth, and so the patients essentially survive better. If there are epistatic interactions, that would be a third reason why they again actually could be, fitted to the data. And then the question is, how can you actually find the real reason behind a certain behavior?
Absolutely. Yeah.
So how would you approach this then, right?
I agree that the problem with these kind of models, really the fundamental problem that all of us grapple with, is this fact that so many of these parameters are not well understood, and the interactions between cells are not well understood, and also the interactions between mutations are not well understood. And so the aim here was not to, as I say, get exact explanations. It's more to take what we take the simplest kind of null model one can think of and see if that's already sufficient to explain the patterns that emerge, or whether we need something else. And I think what we show here is that if you only need to assume what we see as the most basic setup of having some spatial structure and having these beneficial mutations and some variation in the driver mutation rate, and already that's sufficient to recapture these kind of patterns. And one can imagine that, yeah, there are many other ways of doing it and so on. And so I think the next step really is to get better data and to get better clinical data and experimental data so that we can narrow down some of these parameter values. Because otherwise,
yeah, like the mutation rates, for example,
yeah,
maybe, right? So that you can actually fix some of your parameters and then investigate other ones.
Yeah, so the approach we took here was just to do lots of sensitivity analysis. So we looked at these very wide ranges, and then we looked at all the possible combinations of these parameter values. So we had to run many cohorts to do that, thousands of simulations, and that's the best approach we could do in the face of uncertainty. But the more of these parameters we can narrow down, the better we can really start to infer some of the other parameter values.
Okay, thank you. And thanks for a great talk.
Thank you.
Great. Any more questions? We have time for more questions. I have one. Rob, it's great to know that you are working on TRACERx data as well. So another major component in TRACERx is also the microenvironment, for example, immune cells. Have you thought about including them in your modeling?
Yes, I've certainly thought about it, and this is another possible extension. I think we list some of these possible extensions in the discussion of the paper. I can't remember if it's in this paper or in the associated preprint, but yeah, I think it would be fascinating, because this is an alternative explanation for these evolutionary modes, is that they're very influenced by the immune system. So the framework that I've really pursued is what are the effects of spatial structure, and in doing that, I don't include any particular immune effects, but I think it would be interesting to see whether various hypotheses about how the immune system is acting could produce similar patterns, and whether we'd be able to distinguish between these alternative hypotheses, what kind of data we need to do that. So that's why I'm very interested in the work that labs such as your own are doing to really start quantifying where these immune cells are and how many of them there are and what they're doing. So yeah, I'm very interested in that.
Yeah, I'll be very interested in learning also how you would be able to incorporate something like this, and what exactly are the type of data you need in order to do that, to define some of the parameters.
Yeah, so I'm in discussion with various people about that, but the consensus seems to be that the kind of data we need doesn't already exist. So it's a question of how do we go about gathering the appropriate data, and how can experiments or assays be designed to do that. And so I would really welcome anyone who has ideas there to get in touch with me, so we can discuss ways forward on that, because I think the models are now more sophisticated than we have data to parameterize them. This is the current bottleneck.
Excellent. Thank you so much, Rob. Any more questions? If not, we'll move on to the next session. Thank you again, Rob, for a fantastic talk.